Now we're going to turn to our keynote speaker, Susan Baserga, and I just wanted to introduce Susan to everyone. So it's really an honor today to introduce our keynote speaker, Susan Baserga. Susan is a William H. Fleming Professor of Molecular Biophysics and Biochemistry at Yale University. Interestingly enough, Susan has spent her academic career studying the role of important RNAs in the biogenesis of ribonucleoprotein complexes. So first as a postdoctoral fellow at Jones Stites, she studied the role of snRNAs in assembling an active spliceosome. And in her own lab, she studies the mechanisms of ribosome biogenesis, and specifically in how snow RNAs recognize and modify ribosomal RNA. More recently, the Baserga lab has focused on ribosomopathies, and which are, as you probably all know, human diseases that arise from defective ribosomes. So it's clearly evident that Susan has made seminal contributions to our understanding of mechanisms of translational control. There are two other noted contributions by Susan that I would like to comment on. And so the first is Susan has a really deep commitment to mentoring the next generation of scientists, and this is exemplified by her impressive track record of mentoring junior scientists. The second contribution is her service to our scientific community. She's the program director of an NIH T32 training grant for graduate students in the cellular and molecular biology program. And she additionally serves as a reviewer on numerous NIH and private foundation grant panels. And so we really thank her for that service, because we need people, and we need people who understand ribosomes on these types of panels. So now I'm going to turn the floor over to our keynote speaker, Susan Baserga, who is going to present her work on unbiased discovery of molecules that control nucleolar function.
Thank you very much, Christine. That was very kind. And thank you also to the organizers. I'm truly honored to be here today. So I'm very excited to present to you our findings on how we discover new molecules that control nucleolar function. But I really wanted to title this talk, How to Find What You're Not Looking For. Because for me, this has been a big adventure. I'm trying to turn off my thing here. It's been a big adventure in trying to find molecules that I haven't already hypothesized are required for ribosome biogenesis. So it's easy to envision that you can find and work on molecules that are in the nucleolus, because of course they'll be involved in making ribosomes. I'm way more interested in finding the ones that are outside of the nucleolus, and that's what I'll tell you about today. So in general, these are the questions we work on. What is the machinery that makes ribosomes in human cells? And we pioneered the purification and identification of proteins in the SSU processome in the early aughts. Since then, we've been working on how does it work. We've recently taken on the pathogenesis of ribosomopathies as a challenge, and we sometimes dabble in using the nucleolus as a target for cancer therapy. So to tell you all, I'll remind you all what the nucleolus is. You can see nucleoli here. These are these phase-separated structures that are in the nucleolus. They're required for cell division in all human cells. The nucleolus very curiously reforms in the nucleus after each mitosis in eukaryotic cells, with the exception of the two yeasts, Cerevisiae and Pombi, where it does not dissolve during mitosis. And of course, the nucleolus is the site of making ribosomes. Several years ago, we wrote a review, and there's also a podcast available called Mysterious Ribosomopathies, where you can see a picture of the ribosome in the midst of translation, interpreted by the science art department. It looks to me, the ribosome is a jellyfish. I just wanted to let you know that we're working on an interesting new ribosomopathy. It's actually the second case of ANE syndrome, which stands for alopecia neuroendocrine syndrome. It's only the second family. And very curiously, this particular child has biallelic splicing variants in a protein required for making the large subunit. She inherited one allele from one parent that's completely null and one allele that's partially functional, and we've probed its function in both human cells and in yeast cells. And that manuscript's in preparation. So our main question is, what proteins regulate nucleolar structure and function in human cells? And to answer this question, we carried out a genome-wide high-content RNAi screen, which we did several years ago now, and published it in Cell Reports in 2018. And this was the work of Kat McCann and Katie Farley-Barnes, who studied this side of the screen, and that is, in the cells we work in, we have 2 to 3 nucleoli per cell, but they studied what happens when you deplete proteins and come up with 1 nucleolus per cell. Lisa Ogawa-McLean is working on this side of the screen, where you have 5 or more nucleoli per cell, and she's very curiously finding that most of these hits have to do with ribosomal RNA transcription, otherwise effectors of Pol I transcription. Reminding you about how many nucleoli you can have, that there are 10 sites of the rDNA in human diploid cells, chromosomes 13, 14, 15, 21, 22 on the little P arms. But most of the times you don't see 10 nucleoli in cells. You see an average of 2 to 3. And this you can see in this very first panel here. We're standing with the antifibrillary and antibodies with high throughput analysis using cell profilers, seeing you have about 3 nucleoli per cell in MCF10A cells, which are the breast epithelial cells we use in our studies. We looked at about 10 different cell lines. I'm only showing you 3 here. HeLa cells, which remember are not diploid and they're aneuploid, also have 2 to 3 nucleoli per cell. U2OS cells, which is a colon carcinoma cell line, curiously have an average of 5 to 6, 6 to 7 nucleoli per cell. And these are also aneuploid. And we looked at dog cells, we looked at hamster cells. It doesn't seem to matter species, how many nucleoli you have. I have to tell you up front that we don't actually know what dictates how many nucleoli you have in the end, but we do know that it differs. So our big question was, do proteins outside of the nucleolus of the nucleolus regulate human ribosome biogenesis? And this is how we found the answers, with an unbiased genome-wide screen for proteins required for nucleolar function. So it turns out that in MCF10A cells, which are breast epithelial cells, they are not breast cancer cells, that nucleolar number reports function. So when we disrupt nucleolar number in MCF10A cells, phenotypically, in a dish, under a microscope, we can predict that we will see biochemical defects. And so we carried out a high throughput image-based genome-wide screen to discover these proteins that regulate nucleolar number in these MCF10A cells. And then we did various biochemical things to determine the mechanism of how the hits affect nucleolar function. And these are our screen workflow and our screen statistics. The siRNA screen we used SI-genome from Dharmakon, screened about 18,000 genes. We had a number of hits that we filtered through a series of steps, both viability and expression. We filtered them also by localization. We only had 38 nucleolar ones and 101 that were not nucleolar. And since these were the ones I was most interested to begin with, I was very happy to see that. We chose 43 hits for further validation by siRNA deconvolution. 40 of those validated, and of these we chose 20 for mechanistic assays. And just to clue you in on how you should evaluate these screens when people talk about them, you should always ask people what their Z primes are, because Z prime is the separation of the controls. It tells you whether you can really do the screen or not. And we had excellent Z primes over 0.5, which means that we had excellent separation of controls and excellent screening conditions. These are actually the plate by plate statistics, and you can see what the variability is. So we ended up with diverse regulators of ribosome biogenesis through this screen, 139 hits in the end. When we analyze them in various bioinformatic ways, we can see that they affect all these different pathways. We hypothesized that the reduction in nucleolar number would parallel defective ribosome function. And so we tested 20 of them for function in RNA polymerase I transcription, in pre-RNA processing, or in protein synthesis with puromycin labeling. And you can see that there's 7, 16, and 14 that line up to have defects in those 3 things. So it was true. We could use this screen to find proteins that were involved in ribosome biogenesis. But I wanted to tell you about just one today, and that's the PAX9 protein as a regulator of nucleolar number. these are the data from the screen. This is the negative control. This is the positive control. You can see just by eye the difference between 2 to 3 and 1 nucleus. And similarly, PAX9 was a screen hit, 1 nucleus per cell, and these are the histograms from the hits, the actual quantitation of the data. So PAX9 is an RNA polymerase II transcription factor involved in embryonic development. It actually binds the DNA directly. And it has been found to be required, deletion or mutation in PAX9 has been found to be the cause of a condition called oligodontia, and that is when you lose your teeth, and it's usually of the molars, and this has been found in multiple populations worldwide, so this is actually likely to be true. So because of the connection between ribosome biogenesis and Treacher-Collins syndrome, we were very interested in PAX9 as a potential hit because of the connection with craniofacial, possible connection with craniofacial development. And so we set out to investigate the role of PAX9 in making ribosomes, both in human cells, and we collaborate with Mustafa Koko's library in the pediatrics department at Yale on a frog model for ribosomopathies, and we use Xenopus tropicalis because it's a diploid frog instead of lavis. And so our hypothesis was that PAX9 regulates one or more steps of making ribosomes. And so as you know, ribosomes are made in the nucleolus by RNA polymerase I, and they're transcribed into initial transcript to make eventually the 18S, 5.8S, and 28S ribosomal RNAs. These are assembled with ribosomes and exported to the cytoplasm, where they participate in translation. So we checked whether PAX9 was involved in RNA polymerase I transcription, because we reasoned that since it was a Pol2 transcription factor, perhaps it also was moonlighting as a Pol1 transcription factor, and that turned out not to be true. We did see, however, the depletion of PAX9 results in defects in pre-RNA processing, and I'll show you that in the next slide, I think. And then in the end, we showed that siRNA depletion of PAX9 leads to reduction in protein synthesis. So this is a northern blot of MCF10A cells depleted of PAX9, blotting for precursor to the small subunit, and we can see here that we have accumulation of the 30S transcript, which is this precursor to the 18S ribosomal RNA, and then depletion of its subsequent product, which is the 21S RNA. And we used Dmitry Pestov's RAMP method to evaluate these ratios, because we think it's the easiest way to see these. And you can see that in every case we get accumulation of intermediates that lead to defects in 40S subunit biogenesis. But here we're stuck with a dilemma. We have a protein that's a Pol2 transcription factor, but it's clearly required for making 40S subunits. So how does it do this? And we had to hypothesize that it must do this indirectly by transcription of nucleolar proteins. And so PAX9, we hypothesized that PAX9 must transcribe nucleolar proteins that then go to the nucleolus and participate in ribosome biogenesis. And so then we went on to ask whether we could see if this was true. And we did this by using RNA-seq. We depleted PAX9 and MCF10A cells and asked if we could see proteins depleted as well as PAX9. We found a whole bunch over and under expressed. There were, of these decreased expression, these 1,200 decreased expression, 335 were mRNAs encoding nucleolar proteins, which means that this is overrepresented based on how, the 5% of total proteins you think are nucleolar in the cell. And of these 335, we chose 5 candidates for further analysis. And these candidates are listed here. And indeed, we did choose candidates that we thought would support our arguments. And so we tested each of those candidates by northern blot by siRNA depletion to ask if they gave us the same northern blot results that depletion of PAX9 did, because that would then support our hypothesis that PAX9 depletion led to their depletion, which leads to this a northern blot phenotype that we can detect on these gels, and indeed that turned out to be true. This is depletion of PAX9, and this is depletion of all these factors. These are all small subunit ribosomal components. So of course, they lead to accumulation of precursors to the small ribosomal subunit. Fibrillary is also required for U3 snore and P function, so that's why it does. And we knew that RPL5 would lead to a different result. So this supports our hypothesis. Depletion of PAX9 leads to depletion of a group of nucleolar proteins. Among that group are these proteins. But the next step was to then show that PAX9 directly affects transcription of these proteins. The problem with that is that we didn't have any good antibodies to do a PAX9 ChIP-seq or even PAX9 ChIP. And so we resorted to a poor man's way of doing that. Instead, we did RNA polymerase II ChIP-seq across the genome in a PAX9 depletion and found that while the RNA polymerase II distribution across these genes was reduced, it was not statistically significant, which could just mean that you need a combination of the reduction of these, of expression of these proteins. We did see about 19 proteins that when we do RNA polymerase II ChIP-seq have reduced RNA polymerase II occupancy. 19 nucleolar proteins. We've tested some of those. We only tested a handful, and we did not see this defect. But there's another 15 to test. So we think that the way that PAX9 works is indirectly by affecting the transcription of proteins required for nucleolar function. So, but we wanted to extend these results to frogs because we are mostly interested in the developmental defects that went along with PAX9 deletion and mutation in humans. And so what we do in frogs is, with our collaborator, in this case it was Eng and Denise, we have a control embryo frog, or we knock down PAX9 with morpholino oligos, or with 2 different CRISPRs. And we ask what happens to their faces, and you can see that in every case, the face is disrupted, and what we ended up calling this was a bullet-headed phenotype. It becomes a rounded head and distorted features. And this correlates with defects in making the cartilages of the face, and this is OCT imaging, which allows us to see all the cartilages, including the Meckel's, the cerebrococcygeal, and the gill cartilages, and this is the normal side of things. And this is one of the frogs that has abnormal cartilages. Every single cartilage is abnormal. The gill cartilage is grossly abnormal, as is the Meckel's and the cerebrococcygeal. So depletion of PAX9 causes defects in craniofacial development in frogs. And, oh, and I forgot to say that we can rescue this phenotype by adding back the human construct, which is the way most people do that. They take the human construct, which is not subject to breakdown by the morpholinos, and that then rescues the craniofacial phenotype. And we also asked whether we can get a defect in ribosome biogenesis on a northern blot from embryos that were depleted of PAX9, in this case with the PAX9 morpholino oligo. And frog RNA was harvested and run on northern blots and then analyzed, in this case, for precursors to the small subunit. And we do get defects in making the small subunit. The particular defect we get here is cleavage at site 2, which leads to a reduction in levels of the 36 ribosomal RNA precursor and accumulation here of this 18S.5 precursor, which is right here, which is both are consistent with defects in site 2 cleavage, so approximately correlating with what we see in human tissue culture cells. So that told us that our new screen for protein determinants of nucleolar number reveals multiple levels of regulation of ribosome biogenesis in mammalian cells, including by PAX9, but we're also working on others. We have a good lead with prolactin, which you think of as a pituitary hormone, but it's also actually made in the breast. And it looks like prolactin is actually a transcription factor for RNA polymerase I. And my graduate student, Cecilia Harold, got a NASA grant actually to study this. So begging the question, are there other molecules required for nucleolar function? So I told you about our genome-wide screen, and the analysis for the mechanism of how these work is ongoing. And the PAX9, which we just published 2 weeks ago in PLOS Genetics, is just one example of these. Prolactin is another. So in progress are these other very exciting things that are all at different states, and I'm only going to tell you about one, but to tell you that we're in hot pursuit of the others. So I'm going to tell you about how microRNA mimics affect ribosome biogenesis, but we also bought the library for sponges, microRNA sponges, and we've also bought the library and carried out the results for long non-coding RNAs, and have those results as well as the results for small molecules, which is where our work impinges on potential cancer therapy. So my student Carson Bryant carried out a high content screen with microRNA mimics to reveal novel regulators of ribosome biogenesis, again using MCF10A cells. We do all these by reverse transfection. This is a library from Dharmakon that has microRNA mimics, and what that means is that when you put them in cells, they express the microRNAs, and we chose to do these first because we thought that we could best explain how they work. So a microRNA mimic should then target a messenger RNA and inhibit its expression, and that we can interpret based on what we know from our genome-wide screen. So we did this first, and what we do is we just count the number of nucleoli per nuclei to count our hits. And he carried out the screen and discovered a number of microRNA hits shown over here with excellent Z primes. And then he did a secondary screen to look at to see whether they affected RNA polymerase I transcription using 5-ethyluridine click chemistry and a fluorophore. So it worked like a charm. We did a screen for microRNAs that affect nucleolar function, and we counted the nucleoli to find the hits. This is what a non-targeting siRNA looks like, and this is what a microRNA mimic hit looks like for the 1 nucleolus, and what it looks like for the 5+ nucleolus side. This is the double flashlight plot for the 64 microRNA mimics that cause a decrease in nucleolar number from 2 to 3 to 1. And this is where we called the hits, and we said that we had 64 hits, and it was 2 standard deviations from the mean percent effect we called a hit. And on the other side, we called this handful of microRNA mimics hits for the 5+ nucleoli side. And as expected, and this is what's happened with the genome screen, we get many more hits that cause the 1 nucleolus phenotype than we get the 5+ nucleolus phenotype. So we're trying to port some of the assays that we use to high throughput assays so that we can do this faster. And so Carson was finally able to do that with the RNA polymerase I transcription assay. We need to be able to ask if when we express these microRNA mimics, is RNA polymerase I affected? And the answer is yes, and these are preliminary results still. So most microRNA mimics inhibit RNA polymerase I activity. This is activity. There's only two reps, and that's why you see only error bars for 2 reps. But most of these I don't know exactly where I'd break it here, but most of these affect RNA polymerase I transcription. You can see that almost all the 5+ hits affect RNA polymerase I transcription. And so we're in the process of figuring out how this happens. Fortunately, we can do a lot of bioinformatics with the microRNA mimics, because for about half of them, the targets are known, the targets are established, not just hypothesized. And we can see that many of these targets are nucleolar, and now we just have to ask the question, can we further break those down into nucleolar required for Pol I transcription? And that's what Carson's in the midst of doing. So the take-home message that I'd like to share with you is that these unbiased screens can reveal all kinds of molecules, and I showed you about proteins, RNAs, small and large RNAs and drugs required for making ribosomes in the nucleolus. But the challenge, of course, is determining the mechanism. And I showed you what we did for PAX9. We're doing slightly different things for prolactin. and entirely different things for the microRNA mimics. Cecilia Harold has good results with the long non-coding RNAs. She has almost 190 long non-coding RNAs that cause the one nucleolus phenotype, somewhere around there. But we have to validate all of those with a different method, and that's what we're in the process of doing right now. So just to thank everybody in my laboratory who did the work. I've tried to go through and thank people as I've gone through, and of course I'd like to thank the funders who funded this work through the years, and I'm happy to take any questions.
Thank you, Susan. We have a few questions, so we can start. So from Anne Catherine Pratz, she asked, have you looked to see if circular RNAs are also involved?
In ribosome biogenesis, no, I have not.
All right, that was easy. Second question is from Craig.
You can tell me how to do it, and maybe I'll do it.
Gives you something to think about.
Yeah, all good, all good.
So another question is from Craig Kerr. He wondered if the number of nucleoli in a given cell may change over time, and have you tracked cells using live cell imaging and seen this change?
So no, and it's funny, I think about this all the time. I'm dying to do live cell imaging. I'm actually dying to do it for nucleolar formation post-mitosis, because a lot of the hits we get are hits that I would think are involved in that process. And so I have not done that, but I think it's a great idea.
Okay, so another question is from Dominique Wheel. She asked, for microRNA mimics, are the concentration in your screen, is the concentration in your screen what it would be in normal cellular concentrations?
So probably absolutely not. We had to choose an arbitrary concentration. If I told you, I'd be wrong. I knew at one point, I just can't remember. But I think I feel okay about it, because we only got a couple hundred hits, and all the other 3,000 of them were negative. So, and remember, we're doing a weird thing. We're expressing microRNAs and MCF10A cells where they might not normally be expressed at all. So it's a really artificial system.
Another question is from Dmitry Pestov. He asked, from your positive hits, do you see any proteins that could affect the nucleolus indirectly, say a ribosome biogenesis, e.g. by affecting liquid phase separation properties?
I don't know. If you tell me what those are, I can look for them.
That might be something good to do in the Slack offline. So, please do take a look. Oh wait, there's some other ones coming in. I think we have a couple minutes before Rachel may tell me to stop. So another question from Scott Blanchard is, how rapid are the observed changes observed in your screening assays? And he said he's sorry if you missed that.
Yeah, no, no, it's not that you missed it. It's that I probably forgot to tell you. So we actually spent a lot of time trying to figure out how to do the screen when we did it in the first time. And we looked at nucleolar number 1 day, 2 days, and 3 days after depletion. And our Z primes were the best at 3 days, which means that we need mitosis to get the effect that we're trying to see. So, so unlike others who've done these kinds of things, we actually need cell growth and mitosis in order to see what the effect on numbers.
Okay, and another question is from Swati Gakwak. I hope, I'm really sorry about that. Have you checked if AGO2 or any other microRNA machinery protein affects the nucleoli?
I think other people have. I'm pretty sure other people have, but I'm going to Google that after this. I'm pretty sure other people have. Okay, and then this is the last question, I think, because I think we're almost out of time. The question is from Davide Ruggiero. He asks, is PAX9 overexpressed in cancer, and is its expression changed during cell cycle phases? So, Davide, I don't know the answer to the question. I don't think anybody's looked. PAX9 is the most understudied of all the PAXes. So I'm pretty certain it's probably will not be overexpressed in cancer. And what was the second part, Christine?
Sorry, I already moved it.
Expression varied during.
Oh, expression varied during the cell cycle. Oh, you know, we didn't look, so I don't know. PAX9 is involved in Wnt signaling, and actually, when we found when we looked at the RNA-seq data set, we could see the Wnt signaling effects in the RNA-seq data set. So that was the one thing we found that others have found. Thank you, everybody.
Great. Yeah, I think thank you very much, Susan. That was fantastic. And maybe I'll turn it over to Rachel, who may have more information.
I have nothing else to say beyond thank you so much, Susan, for sharing that story with us. That was great. And to remind everybody to take advantage of the poster sessions. And our last day starts again tomorrow at the usual time.